During the course of the talk, you can ask questions using the chat panel on the right-hand side, and there will also be a Q&A session at the end of the talk. So please go ahead and ask your questions there, and we'll answer all of your questions then. Third, we have an active poll running right now with 3 simple questions. Also on the right-hand side, please kindly help us fill it out. And lastly, there will be a post-webinar survey, so if you can help us fill that out, that will also help improve our future webinars. Okay, so our speaker for the day is Nicoya Maheshwari. She's the APAC and Rest of the World Business Development Manager at Nicoya. I work with Nicoya almost every day now, and I have to say she's absolutely an expert on the SPR. And her background in nanoengineering and life sciences make her both really great at supporting the instrument itself, and she will talk to you about the many applications of the SPR system. She has traveled the world to demo Nicoya's SPR systems, including in Vietnam and Thailand, and she'll be very soon coming to Singapore as well. So without further ado, I'll pass the rest of the webinar to Nicoya now. All right, thanks, Nicoya. Please go ahead.
Thank you, James, and thank you, everybody. It's quite early for a few of you for joining us. So today we'll be definitely talking a lot about Nicoya's SPR instrumentation and how it can help you do your research. So let's get started. So a few different things we'll be discussing today is how antibody screening happens and where antibodies are useful, especially in the focus area with infectious diseases. And then we will go into a few different applications, talking about characterizing antibodies anywhere from affinity characterization for immunoassay development to epitope characterization as well, and working with crude samples. It is to understand what is an antibody and how the current screening process for antibodies looks like. So an antibody is basically a host protein that is produced by your immune system in response to a foreign material that it would see. So this could be a virus, it could be a different foreign material, but when it sees that, your immune system will activate and produce these antibodies. Once these antibodies are produced, they can perform a lot of different functions. They can be neutralizing antibodies. They can be used as therapeutic antibodies, among several other ways. Once you produce an antibody or you have a library of antibodies, the gold classic method of characterizing them to figure out first the hits for your antibodies, so do you see yes or no binding with a certain protein, is done with ELISA. So ELISA is a fast and classic way of doing it, and it helps you screen through a big library or hybridomas of antibodies to narrow it down to a small amount of antibodies that you will further define and characterize for your assay. So your assay could be, depending based on your research, it could have different applications. So when you're characterizing your antibodies, you might be looking at the FAB domains or the FC domain or how tight the binding is. And for that, you will utilize secondary assays. Assays will effectively help you develop several different applications in your space. So working with virus diagnostics, vaccine development, therapeutic antibody development, maybe drug development, along with serological test development as well. So when you're working on figuring out one of these applications, you have your library, you characterize it with ELISA, and then you have a few different secondary methods to characterize your antibodies. And that's where SPR comes in. So the question is, why SPR compared to every other technique on the market? So SPR, or Surface Plasmon Resonance, will help you with real-time and label-free detection of your biomolecules. So SPR helps you understand how quickly your two biomolecules come together, how quickly they come apart, and how strongly do they bang to each other. So you get your kinetics, your on rate, your off rate. So what you can see as an example here is that you can have many different types of samples that can have the same affinity, but depending on the on rate and the off rate of the system, you can actually define if it is a good binder versus a weaker binder. This is very important when you're defining a lot of different antibodies, doing affinity characterization in biomolecular R&D and drug discovery. So the question is, how does this actual detection happen? So quickly going over some basics for SPR, you will have your sensor chip layer, which will help you with the detection mechanism, of course. And you immobilize the first biomolecule, which is the ligand, on the sensor chip. So this will be your stationary molecule. And then you will bring in the analyte. If there is binding, the analyte will start binding to your ligand, and you will see an increase in the response. As the analyte dissociates from your ligand, you will see a decrease in the response. Once you generate 3 to 5 of these curves, you will be able to mathematically calculate the on rate, off rate, and KD by fitting this data. The SPR advantage goes into a lot of options. The first one, it will give you a lot of insight about your samples, the on rate, the off rate, the specificity, the sensitivity. And it can do that for a lot of different types of samples beyond antibodies, proteins, nucleic acids, viruses, peptides. We understand that as you work in a research lab, may it be in pharmaceutical setting, in an industry setting, or in your own lab, projects can change, project scopes can change, and SPR is able to accommodate that by being able to work with a lot of different types of samples. You can do work with assay automation, so a lot of the injections will be automated if you wish, and the protocols are simplified for young researchers to be able to work with SPR systems. Overall, you reduce experimental variability. You can work with crude samples. So let's say you're working with serum or plasma or cell lysate, or you're working with supernatant. You can do that with SPR. And then when you're working with the samples, you generally might not have a lot of sample available, and SPR overall requires a pretty little amount of sample, which helps you with taking your research to a larger space. As you're thinking about SPR, a few different things you can do with SPR is screening, affinity and kinetics, characterization, of course, epitope mapping, concentration analysis, understanding if temperature varies your binding, which will fall under thermodynamics, and competition assays. So off today, I will be talking about the OpenSPR, which is our benchtop system from Nicoya. As you can see in the little image here, it is the size of a household microwave, so it does not occupy too much space on your lab bench. It is exceptionally user-friendly. Anybody in your lab can be trained to use it. You do not need a dedicated technician. You're able to achieve real-time data and analyze it while you're receiving the data, and alter your assay as you require to. And it's a fairly low-maintenance instrument and just requires regular cleaning, and that's all. So let's dive into some of the applications that we have performed with the OpenSPR. First off, with the bread and butter of SPR being kinetics characterization. So the example I have here is from one of our application notes where we have immobilized a mouse IgG, and we're trying to study the binding of the mouse IgG towards a few different analyte options. Our negative control is the human IgG, which should not bind to our ligand. And for the analytes, we have a positive control analyte, which is a purified sample. And then we have an analyte, which is present in serum. So what we can show over here is on the top left corner, we have immobilized our mouse IgG on our carboxyl sensor. And when the human IgG is introduced, which is a negative control, it does not show any binding, which is what we expect it to be. So that's fantastic. And then So there's no issues with specificity towards the mouse IgG for the human IgG. So that's great. And then on the right-hand side, what you see on the top right corner is we have the binding of the purified mouse IgG, which is our positive control to the ligand immobilized on the sensor. The colorful graphs are the raw data obtained from the OpenSPR, and the solid black lines are the fitted data, which are fitted using the post-processing software using a 1:1 Langmuir model. At the bottom, what we see over here is the binding of the anti-mouse IgG, which was in goat serum at variable different concentrations. And as we analyze the data, both from the purified as well as your complex media sample, the serum sample, we're able to see that we do not see a difference in the kinetics, which is very useful when you're thinking of working with crude media or crude samples. Moving forward, you've characterized your antibody, you've got your kinetics. How can you use it? So one of the examples that we have that we characterize antibodies was for an immunoassay development for SARS-CoV-2. We're all aware of the coronavirus and how it changed our world over the last 2 years. And part of the process of how we were looking at developing an immunoassay, it will be explained further. So what we did over here is the first step for developing an immunoassay is to select a primary antibody that will, or a capture antibody basically, that has a high affinity to its target protein. And in this case, the target protein is SARS-CoV-2. So it is very important that you select a very sensitive and specific antibody that can only bind to the SARS-CoV-2. So as we were doing this assay and we were designing this assay, we purchased several commercially available options for the antibodies. We sourced them, and then we tested them against the SARS-CoV-2 to figure out which had pretty strong, which emerged as strong binders. So as we did this, we coupled the spike protein to the actual sensor, and then we flew the different, we passed the different antibodies on top of the sensor. Once we did that for 3 to 5 concentrations for each antibody, and then we were able to fit the data to actually look at the final kinetics. We clearly had a range of antibodies, and we selected the ones that had tight binding in the picomolar nanomolar range, which you can see on the right-hand side. Of course, it's great to be able to select an antibody that binds to your target, but you want to check if it has cross-reactivity towards other targets as well. So in this case, what we ended up doing was that we, of course, had the antibodies that were specific to SARS-CoV-2, which is fantastic. And then we also tested them against MERS and SARS-CoV-1. And what we are seeing over here, that once we did the testing with SPR, the first 4 antibodies did not show any cross-reactivity towards the MERS or the SARS, but the last 4 did, and they were eliminated from our process while we were trying to discover our specific capture antibody towards the SARS-CoV-2 itself. On the thread over here to trying to figure out how to potentially amplify the limit of detection, or even try to design an assay where you need a secondary antibody, which might lead to a calorimetric test. We now went down to hunt for a secondary antibody. So as we did that, what we ended up doing that among our commercially available options, we first had our primary antibody or the capture antibody on the surface, and we had our protein bind, and then on top of that, we introduced the different antibodies ranging from this SARS antibody 1, 2, 3, 4 as per our labeling scheme. And what we saw over here is just one example where we had our primary antibody showing binding, but also our secondary antibody showing response as well, which indicates that they have different epitopes within the SARS-CoV-2 that they're binding to, which is fantastic. We did that for a range of antibodies. So on the right-hand side, you can see this little table. So some antibodies were eliminated in part 1 of the experiment, so they were not tested. But the combinations that were tested, we can see on the right-hand side, and the light blue sections is what indicates the antibodies that had no epitope overlap, and that's great. And the other ones were decided against because they did show either an epitope overlap, and that would be an issue. As we continue on the path, when you're working with antibodies, you might end up modifying your antibodies for different purposes. One of them could be to couple the antibody in a stable format on your sensor or your immunoassay development characterization piece. So when you're doing that, biotin streptavidin is one of the standard methods to do that. So that's great. So let's say if you were to biotinylate an antibody or buy it from a commercial vendor, both options are available. You always want to check if the biotinylation has changed any of the characteristics of your antibody, if it has blocked the binding site for your actual antigen. So for doing all of that, you can, of course, use the OpenSPR as well. So in this case, if you were, for this case, if you are biotinylating your antibody in-house, which is definitely a lot cheaper than getting commercially sourced options, once you have biotinylated your antibody, what you could do is what we see over here. You've got your spike protein. You bring your sample in, and it shows binding to your spike protein. Perfect. And then to figure out if the biotin tag actually got attached onto your antibody, you can then pass on streptavidin to see if you see a response, which we do over here with the blue signal. So this indicates that, yes, the biotinylation was effective, as well as our antibody continues to bind to our protein or antigen of interest. Now, another piece you can check is if the kinetics have changed at all between the biotinylation and non-biotinylation piece of the antibody. So what you can do over here is you continue to have your biotinylated and non-biotinylated antibody and testing against your antigen. And followed by that, once we defined the kinetics, we saw that there was not a major significant change in the kinetics, which indicates that the biotinylation and the antibody modification has not caused any changes to the affinity of the system. So overall, when you are working towards potentially an immunoassay development, something to think about is all of the work where we did affinity ranking of antibodies that were commercially purchased, as well as modified, checking for cross-reactivity between antibodies and binning them based on the epitopes, as well as producing antibody modifications and checking on them. We did a lot of this work within 6 weeks. And so can you. As we continue to go through potentially a lot of different variants of coronavirus, what we did over here is that we got commercially sourced proteins from Sinobiological and tested them against the antibodies that were previously tested against the first original variant. And what we saw is that depending on the antibody that was there, there may be some changes in the kinetics, but not very much, and we can check for binding as well. When we are working with SPR, may it be Nicoya's instrumentation or others, you're working quite often with fluidics and pumps and valves and tubes. Sometimes, depending on the instrumentation, there can be frequent downtime or expensive to outsource in general. There can be some tedious processes involved if you have to do a lot of maintenance, and you're using very, very high sample volumes and time-consuming optimization. This might be a concern overall. So if you were working with antibody screening on a very large scale, we have our secondary solution, which is the ALTO. Alto is a 16-channel digital SPR instrument that is ideal for MAb discovery, so monoclonal antibody discovery and optimization. What we have over here with the Alto is that we have everything on a cartridge, which I will show you in a minute, and you will have all the sensors and the fluidics and the reagents integrated in one container. It is a benchtop platform, which is fluidics free. So again, the footprint is fairly small, and you can have the cloud-based software or local software as you prefer to access your data wherever you wish. So let me just explain to you how the cartridge works before we go into an application space for the same. So this is the cartridge where you will have 350 nanoliter volume droplets being dispensed and moved across a grid of electrodes. So that's a very, very low sample volume to work with. We only load 2 microliters of sample to the actual wells here. Once the sample is loaded, the software is programmed to create the serial dilutions for you, and you will see that happen on the left-hand side over here, where the droplets are merging and splitting. And last but not the least, once the droplets have lined up, they can actually go on top of your sensor one after the other between the analyte and the droplet to create the association and dissociation phases. So you are working with a 16-channel instrument at this point, which will allow for a much larger impact when you're working with a lot of samples and go a lot faster with parallelization. You only use 2 microliter sample volume. So anybody who's worked with antibody characterization at some point of time in their life would have not had enough antibody volume. And this is where Alto comes into play, because this is a pretty low sample volume to use, and you can perform multiple experiments either with SPR or use your sample for other secondary methods for characterization. You can run crude samples without any issues because there's no pumps and valves and tubes. You can set up your assay remotely if you wish, or you can, as well as the data analysis if you have the cloud mode. And you can test a variety of conditions when you're optimizing within a single cartridge to speed up your optimization process and then get the data you need. So one of the examples I have here for you is Kinetics characterization and epitope binning for the influenza virus. So step number 1, as always, we wanted to do kinetic analysis for the influenza A antibody and the hemagglutinin antibody, which we immobilized directly on our carboxyl cartridges. Once these were immobilized using covalent coupling, we were able to introduce our analytes respective of the antibodies and perform a kinetic analysis to figure out the kinetic constants. So what we see over here is there's a few different ways you can always perform your experiment. You can do single sequence kinetics, or you could do multi-sequence kinetics. So if you're doing either of the two, Alto will be able to support them both. But the big deal here is that you have your automated serial dilutions, which are able to introduce the 5 different concentrations you're looking for and give you the kinetic constants that you're looking for. We did this for several different antibodies, of course, which was fantastic. We were able to do a lot of this work really fast because everything was parallelized. And we were able to affinity rank bodies based on the kinetic results we obtained. Continuing down this path, we again wanted to look at epitope binning. It's a big application in the antibody space. So what we did over here was we had our surface influenza A antibody coupled to the sensor, followed by introducing the influenza A nucleoprotein. And then once this was optimized, we were able to introduce different solution antibodies. So an example here is what we see is that if you have the binding of the 2 antibodies on 2 different epitopes, you should see a signal change on both samples. But what we see over here is that the solution antibody NP3, we can see a signal rise. So we can say it's different than the capture antibody in terms of the epitope, which is great. But then you've got your solution antibody NP2, which was clearly competing for the same epitope as the capture, and we see no signal now. So using this methodology, we were able to do this for a 5x5 epitope binning system, where we had 5 different surface antibodies and 5 different solution antibodies. And when we did this, we were able to characterize if the antibodies had the same epitopes or the different epitopes for binding onto the antigen of interest. And this is how we could differentiate pairs of antibodies that would work for the next step of the assay. Overall, a lot of this work was done within a week. The amount of reagents used was very, very low, which is actually very helpful when you're trying to repeat your experiments or you're trying to run a lot of different experiments. You can broaden your scope, working with purified samples, but also with crude samples, and overall maximizing uptime. Just in case you decided to use a sample that could clog the system, no problem. Remove the cartridge, get a new one, and you're good to go. But thank you all for listening to me talk about how we can work with Nicoya's SPR instruments for antibody engineering. And yeah, off to James for next things.
Yeah, thanks so much, Nupur. So we'll move on to questions and answers. I'm going to wait a little bit to see if any attendees would like to ask questions. Again, you can use the chat function or you can use the Q&A function as well. Feel free to ask Nupur any questions or also any questions for DKSH. Nupur, just wondering, can you expand on the footprint Nicoya has now with your instruments and also the publications?
Absolutely. So Nicoya, we actually celebrated our 10-year anniversary earlier last week, which has been fantastic. We have about 450 plus instruments worldwide, over 30 plus countries, which is fantastic. We have over 175 publications at this point, ranging from anywhere in infectious diseases, virology, bacteriology, immuno-oncology, neurology. So there's a wide range of work that researchers use the OpenSPR and the Alto for. So yeah, that's our global footprint, and we continue to expand with both our product lines, catering both to individual researchers, core lab facilities, as well as pharmaceutical companies.
Question: are your sensor chips reusable, and can they work with other brands?
That's a great question. So our sensor chips, as long as you leave them installed in the instrument, you should be able to work with different kinds of analytes against the same antigen. So let's say you had covalently coupled your ligand on the sensor, on a carboxyl sensor, and you wanted to test 5 different antibodies. You can just keep removing the antibodies, bringing new ones in. Now, if you were working on a capture mechanism like NTA, then you could capture different ligands and analyze different analytes. Can they be used with different brands? This is unfortunately not possible. Our sensors are patented technology, and that's what the whole brand is built about. So no, it cannot be.
Okay. Next question. What is the sensitivity of the OpenSPR instrument and Alto instrument?
So The sensitivity is pretty high compared to as you're working with SPR. So we are working with localized surface plasmon resonance. So we are looking at about 6 RU per picogram per millimeter square, if that's sort of the number you're looking for. But overall, we are able to compare our instrumentation with other leading instrumentation in the market and get similar kinetic values, as well as work with different types of samples, if that helps.
Do you have experience working with antibody drug conjugates?
So there are customers that are working with those kind of systems. So depending on how your experiment, what your samples are, there should be a way to design the experiment to work in your setting. And that's probably something we could discuss definitely offline outside of the webinar, so we can help you one-on-one characterize your assay.
Okay. Are there any other questions from the poor? Okay, if there are no other questions, we can move on to the last slide. Okay. Yeah, again, thanks, Nupur. Just a bit at the end, I want to introduce DKSH for those who are not familiar. DKSH is a market expansion services company, and we're serving primarily the Asia Pacific market. Our strengths are in biophysical and biomolecular characterization. We also have a portfolio of materials characterization, life sciences, and food and beverages business. As I said earlier, DKSH is the exclusive distributor and partner for Nicoya in the Southeast Asia and Taiwan region, which everyone in this webinar should be. If you have any further inquiries about the OpenSPR or the Alto, please get in contact with one of our executives or colleagues shown on this page, and we will be happy to help you further evaluate the SPR for your application. And I want to give thanks to Nupur for your time again. I know it's quite late over there. I really appreciate you taking out your evening and promoting antibody engineering. You've given us some good examples and on how this can help us with our research. And I also want to give thanks to Irene Chen and Bernadette Lim for coordinating this webinar. So thank you, Irene and Bernadette. And lastly, thank you, the audience, for attending. I hope this webinar has been useful for you. And hopefully you've seen how the SPR technology can fit into your benchtop and accelerate your research workflow, either the OpenSPR or the Alto, in whatever biomolecular therapeutic area that you're working in. And lastly, I want to mention that we'll be sending over the slide deck that was presented today to you and the recording to your registered email. Okay, so we'll be ending this webinar now. Thank you very much for your time and have a wonderful rest of your day. Bye-bye.